Hello and welcome to Phacilitate Exchange. Thank you for joining us. Are induced pluripotent stem cells about to experience a renaissance in their application in cell and gene therapy products? That's what I'm going to be exploring today, so please do stick around. Induced pluripotent stem cells have a number of uses. In the past, we've even had conference tracks dedicated to how iPSCs might be used in disease modeling, in preclinical research, for example. And today, I'm joined by two experts whose company, RHEINCELL, have made some significant advances in using iPSC cells as a starting material for cell and gene therapy products. RHEINCELL Therapeutics, who are based just south of Dusseldorf, develop GMP-grade iPSCs. And I'm joined today by Boris Greber, the CSO and head of production, and Thomas Marx, the head of business development. Hello, Thomas. Hello, Boris. How are you?
I'm Michael. How are you doing?
Michael, thanks.
Good.
Doing good.
So first question over to you guys. Can you tell us a little bit more about RHEINCELL Therapeutics and what was the driving force behind starting the company?
Good question. Again, thanks, Michael, for having us. What can we talk about RHEINCELL Therapeutics? As you may know, the company was founded approximately 3 years ago, and it was founded based on the expectation at that time that iPSCs and cell therapies might become a very important element in the development of future therapies. The founders had a very strong network with the cord blood bank, and the idea was really to generate high quality cellular starting materials derived from those sources, from these origins. And that's how we set up the company.
Yeah, exactly. I think just to add on that, I think that's this bringing together these two fields of iPS technology and the cord blood area. This is what makes the company quite unique, and also the fact that we have our own clean room facility here in the building. So I think we're really trying to bring these things together here.
Nice. And I'm going to just jump right in with my sort of big leading question. Are iPSCs about to see a comeback?
Is it a comeback? I'm not sure. I think what we really see is a continuously increasing demand for iPSCs in the market. And since we started our commercial activities back in early 2020, we really can say there is a tangible increasing number of requests we get every other week from customers all around the world, asking for those cells, for our cells, to support their clinical strategies.
Yeah, and maybe just to add on that, I've really spent years in research before joining RHEINCELL and really seen the field rising. And what I can say is simply that there has been enormous progress in iPS technology, and it had simply took a lot of time and years of research to get to the point where we are now. I think now it's really the time to start really working with these cells and really try to implement them in a clinical setting, just because the methods that you need for that are simply worked out by now. Culturing the cells is now easy and safe. Differentiating the cells is it's certainly not easy, but there are numerous really well worked out protocols nowadays, that allow you to differentiate your cell type of interest at an efficiency of, let's say, 80-90%. And certainly also materials that are required for GMP compliant manufacturing are becoming available now. So I think it's really now the time to get started with the clinical applications, and it just took a lot of time, to get to this point. And so we think, this is really the right time to think about clinical applications.
And with that timing, I think what's really interesting is that it feels like it's coincided with a growth in the application of allogeneic cell therapies. I'm interested in your thoughts on the evolution from autologous to allogeneic cell therapies. And do you think that one day allogeneic will replace autologous cell therapies?
Should I first, Thomas? Also, I mean, personally, I think the autologous approach is certainly the more elegant one. You know, it's super elegant because, you know, you would take the, you know, cells of your own body, you know, reprogram them to iPS cells, generate then the tissue of interest out of these cells, and these are really, you know, personalized cells theoretically, right? I mean, this is an elegant approach, no doubt about that, but it's certainly also a super laborious one, and therefore also a super expensive and time-consuming one. So I think ultimately we just need an allogeneic approach, you know, to you know, to reach a broader perspective really for these applications. And that of course then also, you know, implies, you know, developing a concept of how to deal with the issue of immune suppression, right? I mean, this is it's not just about, you know, generating a GMP iPS cell and really, you know, applying it in an allogeneic manner, but really also developing the concept for, you know, immune suppression, or let's say reduction of immune suppression.
Yeah. I mean, Michael, to answer your question, of course, we hope to some extent it's going to play the allogeneic strategies are going to play a higher and more important role, because fundamentally that's what we try to support with our starting material based on Hla homozygous donors. And from a broader perspective, I think on a long term, a autologous cell therapy from a cost perspective will be quite challenging to serve a broader population of patients, both commercially but also from the process perspective. And in addition, of course, we need to, from what we see today in discussions with customers, there is quite some regulatory challenges that need to be overcome if you think about allogeneic strategies. And I think it's a learning market at the end, where the vendors, where customers will need to jointly get to a shared understanding what really makes sense.
I see. And yeah, in terms of, yeah, we've seen some real success stories in autologous therapies recently, and only last week we've had another approval in the autologous CAR-T space. You mentioned that some of the challenges around allogeneic therapies being more widely adopted, you know, being regulatory considerations and so forth. What else do you think needs to happen in order for allogeneic therapies to kind of be the sort of standard approach?
Yeah, I mean, you mentioned the you know, CAR-T cell therapies, perhaps also other, you know, related cancer therapies based on autologous cells. Well, I mean, the difference here is, of course, I mean, well, I mean, the point here is really that you're talking about a personalized therapy. That's true. It's, you know, a personalized cell therapy, although in this case, probably not a stem cell therapy, right? I mean, but it would be highly attractive, I think, to really translate this idea of, you know, cancer therapy to an iPS-based approach, you know, and using stem cells really for that. And I mean, the big advantage there would, of course, be, and that would imply an allogeneic approach, is, you know, you could really generate an indefinite resource of cells, right? I mean, you would only have to manipulate the cells once. You could grow up the cells at the stem cell level to high numbers. You could differentiate, you know, the gene modified iPS cells into the cell type of interest, such as T cells, which could fight a solid tumor later on in patients. But you can make high numbers of these cells, and you can cryopreserve them in multiple units, you know, and really be able to distribute them to various clinics and really serve many patients, you know, with just one cell line potentially, or with just several cell lines. You know, I think that's the potential really of the allogeneic approach, that you know, you can serve many more patients and in much shorter time with Phacilitate, simplified logistics. And so I think that's going to be exciting to see, the field rising also in this direction, not just in the context of cell replacement therapy, which is of course the other big area for the iPS cells, but also in the area of cancer therapy, when it comes to really using gene modified differentiated T cells, for example, to fight solid tumors.
Yeah, and when you speak about allogeneic, I mean, what is also in our awareness getting increasing attention is really using gene editing technologies to modify the corresponding gene pool, which is responsible for tissue rejection, and from this angle try to influence positively and minimize the unfavorable effects. And that's clearly something where we see in the last, just in the last 6-7 months, and more customers who add that element to their strategy.
Oh, brilliant! And it sounds like it sounds to me like there's a camp that are brought into everything that we've talked about so far in terms of the value of iPSC cells. Why don't you tell us a little bit more about RHEINCELL Therapeutics' role in the cell therapy landscape? And we've talked here about starting materials is really where you play. Why don't you tell us a little bit more about the role that RHEINCELL specifically play in this space?
Well, it's definitely, and I think that is something we are really proud of. A critical element in the evolution of RHEINCELL as a company in the past few months was clearly the final certification of our facility to manufacture, and the manufacturing authorization to produce our iPSCs according to the GMP standard. That was a milestone, because now we can provide customers really with cellular starting material that they require to initiate their clinical programs, and to establish such a environment, such a very highly regulated environment for a startup company like us is, of course, quite a huge step to take. It's a long-term process which requires a very significant degree of engagement from all the people involved. Setting up the process, setting up the QA/QC environment, and making sure that we have the right manufacturing facilities around us. It's a huge also financial risk that our investors are taking. It's because we are still talking about a technology which to some extent is at its infancy, right? So you never know where finally it's going to go, and hopefully they will get the return of the investment, but it's a bet. So we are quite happy on that, and with what we have now, which is our iPSCs, GMP-grade, off the shelves. We have a number of lines ready already. We want to play a significant role in that technology globally as a provider of that material.
Yeah, exactly. I mean, at least that's also something we're learning from our contacts, to clients that there's a huge demand really for GMP grade iPS cells that can be exploited commercially. I mean, this is also an aspect that's quite unique about our cells. I mean, we have a full reconsent, from the donors to exploit these cells commercially, and ultimately a therapy has to be a commercial enterprise, right? So I think this is currently the role that we can play, really, to provide applicants, clinicians, biotech companies who are developing cell therapies on the basis of iPS cells to provide these players with a key starting material. And I think there's a huge demand for this. This is what we're learning right now. But beyond that, of course, we want to develop further. We want to play a role, an active role ourselves in offering, providing cell therapeutic approaches. Ideally, providing the tissue ultimately that is being transplanted into the patient. So this is really our long-term vision. But in the during that upon building that up, of course the provision of the iPS cells is certainly a good interesting business case for us as well. And as I said, it's absolutely important, I think, to have a supplier there for these kind of cells.
Yeah. And if you look at what our customers are doing today, Michael, they are looking at getting their process developed quickly, secure, no risk. That's the key aspect, and within hopefully quite a reasonable financial environment. And that's what we try to support. We have cell lines available which are non-GMP, exactly the same as our GMP lines, to support the first evaluation phase, the first testing phase in a commercially feasible manner. And then customers don't need to redo their experiments again when they move to GMP grade. They can immediately switch. And that's where we really think we can add a value. And then providing the GMP lines fully characterized, as Boris said, commercially completely back up, so they can be used for commercial processes by our customers, embedded in the documentation from the donor to the final product, and hopefully acceptable in all major geographies. This allows our customers to focus on their part of the process, develop their tissues, develop their therapies based on something which they know is good and which they can use.
Yeah, I think so too, Thomas. I mean, sorry, just to add on that. I mean, I think really that's the key point, right? I mean, you want to put your efforts really on what matters at the end, and that's the generation of the differentiated tissue, and not so much on the starting material. I mean, we've gone through a really lengthy process of setting all this up, and getting it accepted by the authorities. And I think you really don't want to go through this again, and it's simply convenient, to have a supplier that, provides you with the starting material. I think it's really at least that's what we're learning, really, that there's a demand for this, and
nice.
That's the role we're playing now.
It sounds like a really exciting journey, and you Boris, you subtly mentioned that you've even got some vision of after this phase, that then your role in being the tissue that goes into patients as well sounds that sounds like another really exciting phase for the company. I just want to take a quick step back though, because you've mentioned the donors a couple of times here. Can you talk a little bit about the donors? What exactly is HLA-H, and where do you get your donors from?
Yeah, that's a good question. As Thomas mentioned before, I mean, we are currently focusing mostly on so-called Hla homozygous donors. So these are individuals which you find rarely in the population, but you find them, that basically have identical HLA, cell recognition genes on both the paternal and the maternal chromosomes. And these setups, holds the potential for making iPS cells out of these donors, and thereby being able to match the genotypes of many more recipients than you would be able to do if you would just use an arbitrary donor for iPS generation. And in a cumulative sense, a couple of or several of these cell lines would ultimately be sufficient to match a huge, or at least a significant portion of the population, without the need or for immune suppression, or with a significantly reduced dose of immune suppressant agents, and this is the basis of the approach, to not use gene modification, or not having to use gene modification, because we have access to these privileged donors, which can serve as a cell source, for transplanting many patients, up to 50%, for example, of the Caucasian population with the lines we have currently. And the reason why we have access to this material is because we are basing, the reprogramming process on cord blood. And we're collaborating with a large cord blood archive, and all of these samples in that archive are basically pre-characterized, so we have access to the information. So we have to the genotypes basically of all these donors, and we can basically now cherry pick the archive for these rare cases of Hla homozygosity, and that's the privileged access we have, and we are trying to profit from it and really focus on these special genotypes. Of course, you don't have to follow the concept. You can also just regard ourselves as GMP iPS cells without looking too much at the genotype. But if you wanted to consider the HLA haplotypes in your clinical approach, you would have a nice set of cell lines available.
So basically, a lot of the heavy lifting, all the pre-groundwork has been done by you and the cord blood bank that you're affiliated with, that is then basically saving your clients especially a lot
Absolutely.
of time.
Absolutely, and Thomas mentioned this before. I mean, this was also the what basically the founder of our company here established over many years. I mean, 25 years, I think, of sample collection for the cord blood archive. I mean, this is really what we're basing the concept on now. That's clear. Nice. A long history, actually.
Yeah, and indeed, what we also can say is then you mentioned the HLA aspect, Boris. In addition to that, what we definitely also can say is that the cord blood by itself, of course, it's a neonatal material. So the genetic integrity of the material is superior. It has not seen a lot of exposure to environmental impact at that point. And we've seen that in all the genetic analysis we did for those products. And we see that also in the quality of our final iPSCs, which have if you compare it to what we see in the market so far, have a much lower mutation load and a much higher genetic integrity comparably.
Yeah, that's true. I mean, you can, using bioinformatics tools, and DNA sequencing, you can distinguish, mutations, for example, which were present already in the starting material from mutations which arose, later during the process of iPS cell generation. And when we did that, then we really got this striking result that the starting material that we're using is indeed has got a much lower mutation load than, for example, using adult biopsies, for example, as a starting material for iPS generation. So that was, I mean, we had the hypothesis since a long time, but we've never had a proof, and now we've really generated these data, and it really holds true that there's a lower mutation load in these kind of iPS cells, which I think ultimately improves safety.
This has been really insightful, and I just want to sort of get a sort of strategic picture as well, because our audience are really focused on how they can drive down the cost of goods. And we know that there's always extra complexity when you look at low quality or low consistency of your starting materials. And when it comes to iPSC cells, we don't know any other providers that are dedicated to providing iPSC starting materials. And I'm really interested as to, the companies you work with now, like what would their alternative have been if it wasn't for how you provide that service, and give me a picture of what that service looks like and in helping them on their cost of goods reduction.
Yeah, I mean, it's certainly true that our iPS cells, our GMP iPS cells that we're offering, to clients are not going to be super cheap, but it's certainly because it took a lot of effort, of course, to produce them, and there's a lot of things that you need to consider and obey, documentation wise, the whole process is complex. Requires a clean room facility, and it has to be embedded in a QM system. There's a huge panel of QC assays that you need to perform, and these are costly assays and so forth. So GMP iPS cells cannot be cheap, but it's certainly much more economical, I think, to buy in GMP iPS cells than making them yourselves, because that's going to be much more expensive. So in this regard, I think we're serving a good purpose, and it's definitely much more economical for clients and clinicians to be provided with these cells off the shelf as we offer it than making these cells yourself. I mean, that's just not paying off, I think, financially.
Thanks, Boris.
Yeah, and we mentioned it before. I mean, if you look at what customers are looking for, is they try to avoid redundancy in what they're doing. So being able to use a cell line for your evaluation of your protocol, for your testing, to confirm it works for what you want it to work for, and then being able to just copy paste that into a GMP product in a GMP process is going to save a lot of time. It's a full part of a development process. So that's one where the cost drivers is, I guess. The second cost driver that we see is always when it comes to surprises in quality, deviations. That is simply not acceptable. So having a fully qualified GMP environment, a documentation which is absolutely stringent, is an added value to avoid unnecessary risk and additional cost and time spent to manage deviations. The starting material that we are getting is being processed in an environment which is also a pharmaceutical enterprise. It's a GMP facility, so that's where we are getting the starting material from, and it's part of our entire documentation for the cells. So what customers get is a package. It's a solid, stable product, fully documented, all required backup there from donor to the end product, and I think that's really adding value, and it's, as you said, obviously quite unique so far in the global market.
I'm sensing a sort of predictability as well in if you're using these GMP materials at an early phase, then I feel like, do you guys sense that your clients know exactly what scaling up will look like for them over time, and they can plan around that far easier as well?
Depending on the therapeutic field you try to go for, the requirement for scale up might significantly differ. If you have a therapy field which requires substantial amount of cellular materials of tissue, then you most probably will need to consider different scale-up strategies compared to indications like, for example, in the eye, where the required amount of cellular material will be much less. But what we definitely see is that customers have quite a good idea on what they may need in the future, and they have those discussions with us. It's part of our services, indeed, to also provide our customers with cell banking services for setting up their master cell banks, setting up particular working cell banks for them, to ensure they get more than just one vial with the same quality based on the same standards, and allow them from there to really understand what they need in the future.
Yeah, I mean, talking about scalability, I mean, just from a scientific perspective, I think iPS cells are just great cells to work with, because they grow so fast. They are capable of self-renewal, so they don't age, basically. So scale up with these cells is actually what these cells are made for, right? I mean, you can differentiate them in small scale if you need to, but as Thomas said, some applications require a higher scale. And that's going to be no problem with these cells because they're made for expansion. So yeah, I think that's another aspect why there's so much potential with these cells.
So I've got an example that I'd like to sort of unpack with you guys. And Cytovia recently announced a partnership where they're using iPSC cells or iPSC-derived NK cells for application in solid tumors. We've talked about allogeneic therapies, and yeah, solid tumors is the sort of the major breakthrough that we're all trying to anticipate, definitely in the next sort of 12 to 18 months. And do you think it's partnerships and approaches like this that might help us see that the breakthroughs in solid tumors?
Well, again, maybe I could start from a scientific perspective. I mean, what I can tell you is that our cells have been tested and validated for being able to differentiate along the NK cell lineage. So that is, and also the T cell lineage. So they're definitely suitable for these iPS-based cancer therapies. And as I said in the beginning, I think there's a huge potential, of doing the CAR T cell therapies, for example, or CAR NK cell therapies based on iPS cells, just because you can potentially really serve many patients with just one modified cell line. You can, produce off-the-shelf stocks of the modified cells. You can differentiate these cells in large batches and really treat many patients with just one or just a few cell lines. I think that's really a huge potential, and that's why there's so much interest in taking this approach. And certainly our cells are suitable for these, especially also because they are Hla homozygous, and this allows you then to match the genotypes of the recipient patients as well at the same time.
I fully agree that the opportunity of immune matching is certainly also a very relevant additional aspect when you look at using cellular material to treat malignancies and cancer, in addition to the traditional approach of tissue replacement therapies, where we came from. So it's clearly something where we see and we really anticipate an increasing attention on. Yes.
Thank you. So we're coming up to the top of our interview now. You've talked a little bit about where the company's come from. Last year, you achieved your GMP certification, and we've looked in the future a little bit as well from Boris's vision of how RHEINCELL Therapeutics may develop further. Let's just look in the medium term. What should we expect from RHEINCELL in the next 12 months? Well,
of course, from one aspect which is driving us is be commercially successful. That's a major driver. I think in addition to that, we clearly will work on expanding our technical and scientific profile. We spoke about the aspect of gene editing, the entire service portfolio around the workflow of an iPSC, from developing differentiation protocols together with our customers, bringing those protocols towards a GMP environment, and from there scale it up is clearly elements that come to my mind, Boris.
Absolutely. I mean, sometimes, from a technical perspective, things seem so easy, but then implementing them in a GMP context is much more challenging. And given the experience that we have just made now with setting up the process of iPS generation, I think we really also want to develop further in the direction, as Thomas said, of differentiation and also gene editing, with a focus on adapting these processes to a clean room setting, to a GMP compliant conditions. And I think that's where the demand is going to be, also for our customers, and we are looking forward also to offer partnering services, and to really collaborate with partners on these kind of efforts.
Yeah, and finally, not to forget our ambition to get our own clinical project initiated at some point. That's why we called RHEINCELL Therapeutics. That was one of the key ideas of the founders, and we haven't forgotten that. Yeah.
Nice. It all seems very positive, and it's exciting. Like it feels like you're really customer centric. You really are. The number of times you mentioned your customers and how they have been involved in the growth of the company, and the direction for where you pivot to and how you support them. It's been really exciting to hear. So wishing you the very best of luck in that, and hopefully we'll meet in person in the not too distant future.